The 2024 Nobel Prize in Chemistry, New Protein Design, Innovation, or a Threat to Our Health and Nutrition. Hello friends. In a very recent development, hugely concerning those following the high-fat, high-protein diets, the 2024 Nobel Prize in Chemistry has been awarded to David Baker from the University of Washington and Demis Hassabis, with John Jumper from Google DeepMind, marking a transformative moment in the field of molecular biology. Their pioneering work addresses two long-lasting critical challenges: the design of spectacular synthetic proteins and the prediction of natural protein structures. These breakthroughs actually redefine how we understand and manipulate life's fundamental molecules, with implications that extend well beyond the laboratory. The potential applications in medicine, biotechnology, and more importantly, food science, are staggering, with some pressing questions about how this power will be wielded and what unintended consequences it would bring to us all. One of the discoveries being recognized this year concerns the construction of spectacular proteins. The other is about fulfilling a 50-year-old dream, predicting protein structures from their amino acid sequences. Both of these discoveries open up vast possibilities, says Heiner Linke, chair of the Nobel Committee for Chemistry. David Baker, recognized for his achievements in computational protein design, has spent years perfecting methods to build proteins from scratch, proteins that do not exist in nature but can be crafted to perform highly specialized tasks. His research team's ability to create custom proteins promises revolutionary advances in new medicine development, therapeutics, and environmental solutions, offering new tools to tackle antibiotic resistance, cancer therapies, and some other critical issues. The other half of the prize acknowledges the work of Hassabis and Jumper, who developed AlphaFold 2, an artificial intelligence model that solved the decades-long mystery of protein folding. Yes, protein folding, the process by which amino acid chains fold into complex three-dimensional shapes, has stumped scientists since the 1970s. The folding pattern dictates a protein's function, and predicting these structures accurately was considered one of biology's most difficult problems. The AI-driven AlphaFold 2 has not only cracked this challenge, but also democratized access to structural predictions for millions of known proteins, revolutionizing fields from biomedicine to material science. Proteins, the essential molecular machinery of life, govern nearly every biological function, from enzyme catalysis to immune responses. Keeping this in perspective, these scientific breakthroughs open vast new frontiers, especially in precision medicine. Yet, the potential applications extend further than the healthcare sector. As the tools to manipulate proteins are evolving, they also begin to intersect with food science, pushing towards a future where artificial proteins would supplement or even completely replace our traditional dietary sources. Now, as this Nobel Prize is going well beyond scientific recognition, it signals a fundamental shift in how we approach life's essential molecules. And the question that arises here is that, where do these breakthroughs in protein science take us? Apart from the apparent benefits, what real implications they are to bring to our lives? How will this power reshape our food systems and influence our long-term health, particularly for those of us on the high-fat, high-protein diet plans? As we move ahead in this highly informative video, we'll untangle all these crucial questions, weighing the potential of scientific progress against the uncertainties and disruptions it would bring in our lives. The stakes here are really high for us, and the answers may not be as straightforward as they seem. Stay with us, as this vital info is not available anywhere else on YouTube, and the real story is about to begin. Let's begin with decoding the Nobel Prize, the problem at hand, and the science and impact of protein design. As we all know, proteins are essential molecules that carry out virtually all biological functions, facilitating metabolism, immune response, energy production, and cell communication. However, understanding and manipulating these complex structures has remained one of the most difficult challenges in molecular biology. A protein's function is dictated entirely by how it folds into a precise three-dimensional shape, but predicting this shape from its amino acid sequence has eluded scientists for decades. This difficulty is known as the protein folding problem, first formulated in the 1970s. Despite advances in experimental techniques, such as X-ray crystallography and nuclear magnetic resonance, NMR, spectroscopy, solving the structure of proteins remained a slow and resource-heavy process. Each protein required dedicated work that could take months or years, hampering scientific progress, particularly in areas like drug discovery and enzyme engineering. Alongside the challenge of understanding natural protein structures, researchers also faced the limitation of biological evolution. Natural proteins are the result of millions of years of evolutionary fine-tuning, optimized for the survival needs of specific organisms. However, the functions needed for modern medical and industrial purposes often require new types of proteins, molecules that do not exist in nature. Designing these proteins from scratch presented enormous technical hurdles. Without the ability to accurately predict how sequences would fold, it was difficult to create proteins with desired functions. This dual challenge, predicting protein structures and designing functional proteins, represented a major bottleneck for biotechnology, medicine, and environmental science. In response to these challenges, scientists pursued two parallel paths: improving protein design algorithms to create new proteins from scratch, and developing predictive tools to unlock the structures of natural proteins faster and with greater precision. These efforts culminated in the breakthroughs that earned the 2024 Nobel Prize in Chemistry, awarded jointly to Dr. David Baker of the University of Washington and Demis Hassabis and John Jumper from Google DeepMind. Let's have a detailed look at what exactly this development refers to and what it means for our future. Starting off with David Baker, solving the almost impossible design problem with computational protein engineering. Dr. David Baker's groundbreaking work addresses the challenge of de novo protein design, the ability to create new proteins from scratch for specific applications. His lab developed Rosetta, a pioneering computational platform that simulates how amino acid sequences fold into stable, functional structures. This tool has evolved over the past 2 decades into one of the most powerful resources for protein engineering, allowing researchers to bypass the limitations of natural proteins and create entirely new molecules. Baker's team demonstrated that proteins could be designed to carry out highly specialized tasks, such as functioning as enzymes that break down environmental pollutants or molecular sensors capable of detecting disease markers. A particularly noteworthy achievement was the creation of a synthetic medicine for respiratory syncytial virus, RSV, the first de novo designed medication to enter clinical trials. This development highlights the transformative potential of custom protein design in the field of medicine, paving the way for more effective and precise therapeutics. Beyond medicine, Baker's designs extend into fields like nanotechnology and green chemistry. His lab has produced enzymes that can break down plastics and materials with enhanced properties for use in industry. The power of computational design lies in its ability to solve problems faster and more accurately than traditional trial and error methods, reducing reliance on natural proteins and expanding the toolkit available to scientists and engineers. Moving to the other two winners, Hassabis and Jumper, solving the protein folding problem with AlphaFold 2. While Baker tackled the design problem, Demis Hassabis and John Jumper of Google DeepMind confronted the protein folding problem, a puzzle that had frustrated biologists for over 50 years. AlphaFold 2, their deep learning-based AI model, achieved what many believed was impossible, accurately predicting the three-dimensional structure of proteins directly from their amino acid sequences. This feat was made possible through neural networks trained on large datasets of protein sequences and structures, combined with advanced optimization techniques to generate predictions with atomic-level accuracy. AlphaFold 2's impact has been profound. Since its release in 2020, the system has solved the structures of more than 200 million proteins, providing researchers with insights that would have taken decades to uncover using experimental methods. Scientists have used AlphaFold 2 to study antibiotic resistance mechanisms, model viral proteins, and develop new enzymes for industrial applications. Its open-access model has democratized protein research, making these insights available to millions of researchers worldwide across 190 countries. The predictive power of AlphaFold 2 has revolutionized several scientific fields. In medicine, it accelerates medication discovery by revealing the exact structure of target proteins, allowing for the design of more effective medications. In materials science, it enables the development of custom enzymes for sustainable chemical processes. By solving the protein folding problem, Hasabi's and Jumper have provided a tool that streamlines research and innovation across numerous disciplines. Talking of the combined impact, unlocking new frontiers. Together, the work of Baker and the DeepMind team represents a paradigm shift in molecular biology. With the ability to design new proteins from scratch and predict the structures of natural proteins with precision, scientists now have tools that dramatically expand the frontiers of biotechnology. These breakthroughs promise to revolutionize medicine, drug development, industrial bioprocesses, and environmental science by providing faster, more effective solutions to previously intractable problems. However, as these technologies mature, they raise new unanswered questions. Custom-designed proteins and AI-predicted structures offer incredible potential, but they also challenge our understanding of biological systems. How will these new proteins behave over time? Are there unforeseen risks in engineering molecules that do not exist in nature? As we embrace these breakthroughs, we must carefully weigh the balance between progress and caution. Moving on, we will explore the implications of these advancements in medicine, biotechnology, and most importantly, nutrition, and see how exactly we are going to be affected, either in a good or a bad manner. This is where it gets crucial for us all, following the diet plans with proteins as an integral part of the whole picture. But before moving to the direct impact on our food and nutrition, let's have a brief look at the other crucial areas. Transforming medicine, precision therapies with engineered proteins. According to the researchers, medicine is one of the primary fields poised to benefit from these breakthroughs. Traditional treatments often rely on broad-spectrum pharmaceuticals that impact healthy and diseased cells alike, leading to side effects and reduced efficacy. With the ability to design targeted protein-based therapies, researchers can now develop precision treatments that interact only with specific biological pathways, minimizing unintended effects and improving outcomes. For example, synthetic antibodies, proteins engineered to bind precisely to disease-related molecules, offer promising new approaches to treating cancers and autoimmune disorders. David Baker's lab has also developed customized immune activators designed to trigger precise immune responses against specific pathogens, including an experimental therapy for respiratory syncytial virus, RSV. These tailored protein treatments are unencumbered by evolutionary constraints, marking the dawn of a new class of highly specialized medical interventions. Plus, AlphaFold 2's predictive capabilities have accelerated the development of therapies by providing accurate models of disease-relevant proteins. This insight enables researchers to identify new therapeutic targets more rapidly, especially in cases where conventional methods would take years. A prominent application involves modeling antibiotic resistance proteins, helping scientists develop new treatments that overcome bacterial resistance, which is one of the most urgent challenges in modern healthcare. Revolutionizing industry, green chemistry, and environmental solutions. The implications of these breakthroughs extend beyond healthcare, reshaping industrial biotechnology and green chemistry. Enzymes, which catalyze essential chemical reactions, play a crucial role in numerous industries. However, natural enzymes often come with limitations in efficiency and stability. Baker's work on synthetic enzymes eliminates these constraints, enabling the creation of highly efficient catalysts tailored for specific industrial processes. One of the most promising applications is the development of enzymes capable of degrading plastics, offering a potential solution to the global plastic waste crisis. These custom-designed proteins can break down plastic molecules at a molecular level, creating opportunities for sustainable recycling systems. Additionally, synthetic enzymes are being employed in biofuel production, enhancing the efficiency of converting biomass into fuel and accelerating the transition toward renewable energy sources. AlphaFold 2 has also facilitated the optimization of industrial enzymes by providing precise models of their structures. This insight allows companies to fine-tune catalysts for pharmaceutical manufacturing, agriculture, and other fields, improving efficiency while minimizing environmental impact. The result is a new generation of sustainable industrial processes that rely less on harmful chemicals and more on biological catalysts tailored for green chemistry. The double-edged sword of precision control. While these innovations hold immense promise, they also introduce new challenges. Engineered proteins and AI-driven predictions can behave unpredictably when applied in real-world environments, raising concerns about unforeseen risks. For example, synthetic enzymes deployed in ecosystems may interact with native species in unexpected ways, while protein-based therapies could trigger unanticipated biological responses over time. Additionally, these breakthroughs prompt critical ethical questions about control and accessibility. As companies race to commercialize these technologies, there is growing concern over how these tools will be regulated and whether they will remain accessible to all or become exclusive intellectual property controlled by a few. The power to manipulate life's molecular foundations must be exercised carefully, with a focus on balancing innovation with long-term safety. Next up in the video, we have something really critical for us, as we will explore how these developments are beginning to influence food science and nutrition, particularly for those of us following high-fat, high-protein diet plans like keto and carnivore, where the tension between natural and synthetic solutions is becoming increasingly significant. The emerging impact on nutrition and food science, a new threat to long-term wellness. The breakthroughs in protein design and structure prediction are now extending beyond medicine and industry into food science, raising serious concerns about the future of human health and nutrition. As these technologies are adopted to create synthetic food alternatives, we must ask whether artificial solutions can genuinely sustain long-term wellness. While these developments offer promise for addressing environmental challenges, they also introduce unanswered questions about nutritional adequacy and unforeseen health risks. The foods we rely on today for essential nutrients may soon be replaced by engineered proteins and fats, and the consequences of this shift could be profound. Several startups backed by Bill Gates and other investors are accelerating this transition toward synthetic foods. Among them, Upside Foods, formerly Memphis Meats, is at the forefront of developing cultivated meat grown directly from animal cells without raising any livestock. With recent funding of $400 million and a valuation surpassing $1 billion, Upside Foods is building large-scale facilities to produce chicken, beef, and seafood, aiming to deliver millions of pounds of lab-grown meat each year. This innovation may promise to reduce the environmental impact of traditional meat production, but this is not exactly the true picture. Remember what we told about Bill Gates-backed projects in our earlier videos, all focused to drive us away from the real nutritional foods. Similarly, Savor, another Gates-backed initiative, is working on producing synthetic fats and butter using carbon capture technology. By transforming carbon dioxide, hydrogen, and oxygen through a thermochemical process, Savor claims to replicate the taste and texture of real animal fats. But again, can this artificial substance match the real produce in any manner? As we discussed in our last video, while these innovations apparently align with sustainability goals, they raise significant concerns about their impact on human health. Natural animal proteins and fats provide more than just calories. They are rich sources of heme iron, vitamin B12, omega-3 fatty acids, and other bioavailable nutrients essential for metabolism, brain function, and immune health. Replicating the full nutritional profile of natural foods in synthetic alternatives is an enormous challenge, as these engineered products may lack key cofactors required for proper absorption. Even if synthetic foods can mimic the structure and taste of natural ones, the long-term bioavailability and health effects remain largely untested. Moreover, the human digestive system has evolved over millennia to process natural proteins and fats efficiently. Replacing these with lab-engineered alternatives introduces the risk of disrupting gut microbiota, which could lead to metabolic disorders and immune dysregulation. Without thorough long-term studies, the cumulative effects of consuming these synthetic products are impossible to predict. Consumers may be drawn to these alternatives for their environmental benefits, but the hidden cost to health could surface years later, catching society unprepared. The increasing push toward synthetic foods raises broader questions about the accessibility of natural foods. As governments and corporations promote lab-grown alternatives for environmental reasons, traditional animal-based foods may become marginalized, either through economic shifts or regulatory changes. If this trend continues, the dietary landscape could be reshaped to favor synthetic substitutes, forcing consumers to rely on engineered products, even if they prefer natural sources of nutrition. This shift would not only limit consumer choice, but could also erode cultural and dietary diversity, as many cuisines and food traditions are deeply rooted in the use of animal products. These developments present a complex challenge. While the promise of technological progress is undeniable, uncritical adoption could jeopardize health outcomes in ways that are difficult to reverse. As we explore these themes further, it becomes clear that rigorous scientific oversight and informed consumer choices will be essential in ensuring that synthetic foods complement rather than compromise human nutrition. Next up, we will explore the exact implications of these developments and assess how synthetic biology can threaten the principles of proper nutrition and our well-being. The game of big risks. Well, friends, the risks posed by synthetic foods, especially those derived from engineered proteins and fats, extend far beyond the superficial concerns of novelty. While these innovations are marketed under the promise of sustainability and progress, they carry serious health risks that remain largely untested. As synthetic alternatives begin to replace traditional sources of nutrition, the long-term impact on human health and biological well-being raises urgent and critical questions. Relying on lab-grown foods and artificial proteins threatens to disrupt metabolic balance, immune function, and nutrient absorption in ways that could cause profound, irreversible damage over time. One of the most immediate concerns revolves around the issue of nutrient bioavailability, the extent to which nutrients are absorbed and utilized by the body. Natural animal products, such as meat, fish, and dairy, provide essential nutrients in bioavailable forms, including heme iron, vitamin B12, long-chain omega-3 fatty acids, and complete amino acid profiles. These nutrients play irreplaceable roles in cellular metabolism, brain function, and immune health. Attempts to replicate these profiles in lab-grown meat or artificial butter face significant limitations. Even if the chemical structure of synthetic proteins mimics that of their natural counterparts, the complex synergies between nutrients, present in real foods, are exceedingly difficult to replicate in artificial systems. Replacing real animal-based proteins with synthetic ones risks introducing metabolic inefficiencies. The human digestive system has evolved over millennia to process and absorb nutrients from natural foods with remarkable efficiency. Introducing synthetic proteins or lab-grown alternatives could disrupt gut microbiota, the complex ecosystem of microorganisms essential for digestion, immunity, and hormonal balance. Disruptions to this microbiota have been linked to autoimmune disorders, metabolic syndromes, and chronic inflammation, conditions already on the rise in modern societies. Long-term reliance on artificial foods, without fully understanding their effects on gut health, could exacerbate these conditions and create new, unpredictable health risks. The long-term health impact of consuming synthetic fats is another area of serious concern. Fats derived from carbon capture processes, such as the artificial butter developed by Savor, may replicate the taste and texture of natural fats, but there is no comprehensive understanding of their metabolic behavior. Natural fats, particularly those from animal sources, contain bioactive compounds, like CLA, conjugated linoleic acid, and phospholipids, that support heart health, immune function, and brain development. Synthetic fats are unlikely to carry these same benefits, and consuming them as a substitute for natural fats could disrupt lipid metabolism, increase the risk of inflammatory diseases, and impair brain function over time. The commercialization of synthetic proteins also presents a monopolization risk. If key technologies, such as those used in precision fermentation and cultivated meat production, remain under corporate control, it could limit public access to nutrient-dense natural foods, consolidating power within a small group of companies. This monopolization of food systems raises the possibility that decisions about what we eat will be dictated by profit motives rather than health priorities, further undermining our individual autonomy and dietary freedom. Well, friends, to put it simple, the 2024 Nobel Prize in Chemistry showcases groundbreaking advancements in protein design and structure prediction, offering extraordinary potential for medicine and industry. Yet, when applied to food and nutrition, these technologies pose serious risks. Synthetic proteins and fats, though marketed as sustainable, come with unproven health effects, metabolic disruption risks, and could undermine access to natural, nutrient-dense foods. While these developments hold immense promise, their misuse in the food industry could very well prioritize profit over our long-term health. Progress must be pursued with caution and responsibility, ensuring that innovation doesn't compromise human wellness. By the way, what is your opinion on this whole development? Would you try any synthetic food in your diet? And how has your experience been on the carnivore diet? What health conditions have you healed so far? Do share your journey in the comments. Press the like button if you found this video helpful, and share it with your loved ones as well. Subscribe now to our channel to get more of such highly informative content about your health and well-being. And if you want to know what special happened 100 years ago related to keto and carnivore, click on the video showing up on the screen to find that out. See you there.